Let me share my screen. So I do hope the slides are visible to all of you. If not, you can let me know in the chat box. So without any delay, let's start. So this session will mainly focus on the question regarding the week 12 lecture materials. Uh, Basically the overall metabolism processes, the oxidation of glucose and the TCA cycles, etc. So let's start with our first question. So this is the first question. It states, which among the following products of glucose oxidation are essential for oxidative phosphorylation? So there are four options: acetyl-CoA, pyruvate, NADH and FeDH2, and the fourth one is NADPH and ATP. You have to identify which among the followings are the product of glucose oxidation that are also essential for oxidative phosphorylation. Select your answer and try to answer in the chat box, and then I will discuss the questions one by one. So there are two processes we are talking about. One is oxidative phosphorylation, another is glucose oxidation. So what is our oxidative phosphorylation is actually, it is basically the process for ATP synthesis that we have discussed in details in the last session. If any of you can answer this question, please do write. Then I will start discussing. So what is our glucose oxidation? Let me write it down. So we have it here. We have our glucose that reacts with 2 moles of ADP and 2 inorganic phosphate with the help of NAD plus, and you end up with 2 pyruvate molecules, and in the process ATP gets synthesized followed by 2 NADH. Obviously the NAD plus is going to be reduced with the formation of water molecules. So this is the glucose oxidation process. So you can already see what are the products of glucose oxidation. You can already see. So which are they? Pyruvate, ATP, NADPH and H2O. Out of them you have to identify which are essential for the process of oxidative phosphorylation. So let me go back one second, because the writing will be shown hampered. So I have already showed you the process for glucose oxidation, and after the formation of the pyruvate, which I just wrote, so basically in the process for glucose oxidation, one second, you have the formation of pyruvate, which is next utilized in the TCA cycle, where from pyruvate you will end up with the formation of CO2. So that is the overall process. So in the whole process of glucose oxidation, you can see out of them NADH, you can see the involvement of NADH and FADH2. Now if you remember the process of oxidative phosphorylation, so this is the overall process of oxidative phosphorylation, where from ADP you are getting ATP, that is the oxidation of ADP to ATP. Meanwhile, reduction of ADP to ATP, and meanwhile this NADH or FADH2 gets oxidized to NAD plus and FAD. So in case of oxidative phosphorylation, we end up with the oxidized form of NADH and FADH2, that is NAD plus and FAD. That means now if this NADH and FADH2 has been oxidized, you again have to reduce them, otherwise this step will no longer occur, because once they are oxidized, if you do not reduce them again, then this ADP to ATP conversion will no longer occur. It will stop up to just this 1 mole of reaction. So to do that, which comes into play, you can look at this overall glucose oxidation and citric acid cycle, where you have the formation of NADH and FADH2. That means these products NADH and FADH2, which are the products of this glucose oxidation, can further help in the oxidative phosphorylation. So the correct answer for question number A should have been option C. NADH and FADH2, these are the products of glucose oxidation that are essential for oxidative phosphorylation. Okay. So let us move to the next question. This one states the combination of cofactors that are involved in conversion of pyruvate to acetyl-CoA. So pyruvate is the result of initial glucose metabolism, where you form this glucose to pyruvate, which is then further utilized in the TCA cycle. For that you need the formation of acetyl-CoA or acetyl coenzyme. You need to identify which are the cofactors that are involved in this conversion from pyruvate to acetyl-CoA. Which are the cofactors that are involved? There are 4 options. If you know the answer, please do write it. Otherwise, I will show it in a minute. Okay, fine. So let us see. This is the pictorial diagram shown on the right hand side, that is for the conversion of pyruvate to acetyl-CoA. Now in this case, in this overall thing, the enzyme that is involved, the pyruvate dehydrogenase, is basically has 3 subunits. One second. So this has pyruvate dehydrogenase has 3 subunits. You can already see E1, E2 and E3. Now what are the cofactors that are associated with these subunits of this enzyme? In case of E1, you can already see there is TPP, that is thiamine pyrophosphate, that is derived from vitamin B1. You already learned about this. So the cofactor that is associated with the E1 subunit of the enzyme is TPP. Fine. Let us look at the second subunit. In the second subunit, we have different modifications of lipoic acids. So lipoic acids get transformed into different process, and that is the cofactor that you can see in case of E2, that is the second subunit. So this is the lipoic acid moieties that you can see, which gets reduced to give you SH groups that further get acetylated. We will discuss this part later in some questions. For now, you can see lipoic acid is the other part that is involved as a cofactor for E2. And in the third case, you can again see this is the pink colored one. This is the E3 subunit of the enzyme, and which one is involved in this case? FAD, which is getting reduced to FADH2. So what are the cofactors we got? TPP, lipoic acid and FAD. So what should have been the correct option for question number 2? Option number D. These are the cofactors that are involved in the overall conversion of pyruvate to acetyl-CoA. Let us move to the next question. It says the proton motive force that is generated by electron transport during oxidative phosphorylation is used for what? First option is create a pore in the inner mitochondrial membrane. induce a conformational change in the ATP synthase, generate the substrates ADP and PI for the ATP synthase, and oxidize NADH to NAD+. So what we are talking about, PMF, or otherwise known as proton motive force. How this is generating? Because in the last session we already discussed that in case of mitochondria, if this is your outer membrane and this is your inner membrane, this is the intermembrane space where you have high concentration of H plus ions, and this is the matrix where you have low concentration of H plus, and you need a transport of H plus ions from matrix to this intermembrane space. This leads to positive charge on this intermembrane side and a negative charge on this matrix side, on this inner membrane, which leads to a electric force. So this is known as the proton motive force, that is arising due to the motion of the protons inside this mitochondria that you can see. So why do we need this in case of oxidative phosphorylation? That was our question. So what should be the correct answer? Ultimately, what does it result? It does induce a conformational change in the ATP synthase. So for the synthesis of ATP, the enzyme that is responsible here in the last step, that is ATP synthase. But for it to attach with the substrate, you need a conformational change, because we have learned it. In some cases, whenever the substrate molecules comes or something, you need a conformational change in the enzyme so that the substrate can fit and give you the desired product. In case of ATP synthase, this proton motive force actually results in the conformational change, which will finally lead to the formation of ATP. Otherwise, it will not form our desired reaction, that is ADP to ATP conversion. So the correct answer for this one should have been option number B. Okay. Hope this is clear. Let us move to the next one. In the fourth question, it is asking that during exercise, when aerobic metabolism cannot keep up with energy requirement, because during exercise there is a deficiency of oxygen in our body. So the aerobic metabolism, which is basically the metabolism in presence of oxygen, that cannot keep up because our body has now deficiency for oxygen. So in that condition, what will happen? There are 4 options that are given to keep up with the energy requirement. So even if we are exercising and there is oxygen deficiency, still we need our body to function. That means our body needs more energy for the function. So how the energy will come? And in that condition, there are 4 options. First one is skeletal muscles will ferment glucose to lactate. Second one is lactate dehydrogenase catalyze the reaction of pyruvate to lactate irreversibly. Lactate is converted to pyruvate for entry into Krebs cycle, and pyruvate is oxidized to CO2 via Krebs cycle. These are the 4 options. So select accordingly which of them will give you the necessary energy when aerobic metabolism cannot keep up. That means basically you are doing in anaerobic condition. Choose your option and then I will discuss. Fine. So what happens in case of our aerobic condition? That is in presence of oxygen molecules. What will happen? In case of aerobic condition, we have our pyruvate, that gets converted to CO2 which is otherwise known as Krebs cycle. But in case of anaerobic condition, that means there is deficiency of oxygen. In that case, pyruvate will be converted to lactate. And how this happens? This happens through the skeletal muscles that ferments the glucose to lactate. Why it is said so? So let me just go back. I don't know whether it is visible. I think the image here is more or less visible. So in case of anaerobic condition, you have your skeletal muscles that will convert pyruvate to lactate, and your NADH is oxidized to NAD+. So that is the process overall known as fermentation. So the overall fermentation process, we can say, comprises of glycolysis plus the conversion of NADPH to NADH to NAD+, that is the oxidation of NADH. This overall process is known as fermentation, and that is done by the skeletal muscles which convert glucose to lactate in case of anaerobic condition. So this was our correct option. Why this one is, second one is not true? If you look at it, it says lactate dehydrogenase catalyzes the reduction of pyruvate to lactate irreversibly. Obviously, lactate dehydrogenase does catalyze this reaction from pyruvate to lactate, that is shown here. But this is a reversible reaction. It is not a irreversible reaction. It will only happen when the oxygen is less. The lactate dehydrogenase is converting pyruvate to lactate. But whenever, suppose you are resting and the oxygen in your body, the level of oxygen in your body is again increasing. In that case, the lactate dehydrogenase will again convert the produced lactase to pyruvate. The reaction is a reversible reaction. It is not an irreversible reaction. So it will again from lactate to pyruvate so that it can go to Krebs cycle. So that is why this last part of the second statement was wrong. That is why option statement number B was a wrong statement. Coming to C, lactate is converted to pyruvate for entry into Krebs cycle. That is obviously not true. That is another step. That is not in anaerobic condition. Again, when you have oxygen in your body, again that will be converted to pyruvate. So that is not something that will happen under anaerobic condition, but that will happen under aerobic condition. So this one was also a false statement. And the final one is pyruvate is oxidized to CO2 via Krebs cycle. This statement is again false because pyruvate oxidation to CO2 happens under aerobic condition, not under anaerobic condition, which I wrote at the beginning of the discussion of this question. So that is why option number A was the correct option for question 4. Let us move to the next one. So the fifth question is, which of the following is or are the intermediates of citric acid cycle? So you know your TCA cycle. Now you need to identify out of these 4 that are written: acetyl coenzyme, citrate, oxaloacetate and succinyl coenzyme. Out of these 4, which are the intermediates of TCA cycle? For that you do need to remember the TCA cycle. This week 12 is mostly memory based questions, and for that you actually need to remember all the glucose oxidation step as well as the TCA cycle or Krebs cycle steps. Try to answer that and then I will show. So let us look at our TCA cycle. I have shown you the answer. So this that is shown in the right hand side is our TCA cycle, where we start from this oxaloacetate and acetyl coenzymes works on oxaloacetate to give you citrate. So citrate is one of our intermediate. We can see oxaloacetate is the starting product, and you have your acetyl CoA. This is the enzyme that interacts with it and forms citrate. So citrate is one of the intermediates. So in the question, this option, that is number 2, is an intermediate of our TCA cycle. Let's move on with this. Next is the citrate to isocitrate formation. Now you have here, sorry, it's not writing properly. One second. So you have the action of aconitase in the next step, where you have the conversion of citrate to isocitrate. Okay, I don't think isocitrate in the option, fine. The next step is formation of isocitrate to alpha ketoglutarate, which is done by isocitrate dehydrogenase. So isocitrate dehydrogenase works in this step, forming the alpha ketoglutarate. Alpha ketoglutarate is also not in the option. The next step is from alpha ketoglutarate we go to succinyl coenzyme. That is done by succinyl coenzyme synthase. So the enzyme that works here is succinyl CoA synthase. With the help of this enzyme, alpha ketoglutarate transformed to succinyl CoA. So yes, succinyl CoA is another intermediate. I don't know why this is not working. So this is another intermediate of our TCA cycle. So yes, fourth one is definitely another intermediate. Next step is succinyl CoA to succinate formation. That happens via the enzyme succinate dehydrogenase. That is not in the option. Then is succinate to fumarate formation. That is done by fumarase. That is also, fumarate is also not in the option. Next step is fumarate to malate, and that happens by malate dehydrogenase. The fumarate to malate, and after that I think that happens by fumarase, I might be wrong. Okay, and the final step is malate to oxaloacetate. That happens via your malate dehydrogenase. So oxaloacetoacetate, this is another intermediate. Why so? Because at the end of the cycle you can see oxaloacetoacetate is being again formed. That is why it is a cycle. So oxaloacetoacetate is again another intermediate of this TCA cycle. However, acetyl CoA, the number 1 option, acetyl CoA, is not an intermediate. It is an enzyme that is functioning on the substrate and the intermediates, but it is itself not being formed in this reaction. The formation of acetyl CoA is completely different, is a series of reactions that are completely different. It is not something that is formed within this TCA cycle. So this is not your intermediate. So 2, 3, 4, these are the intermediates of TCA cycle, not acetyl CoA. So your correct option should be option number B. Okay. Hope this is clear. Let us move to the next one. It says formation of 2 moles of CO2 and coenzyme from 1 mole of acetyl coenzyme, okay, via the citric acid cycle results in the net production of how many moles of, there are 4 options. So for that you need to know the TCA cycle and what are the substrate and what are the products. So let us look. This is the stage 2 of TCA cycle or Krebs cycle, where you can see acetic acid is being converted to 2 CO2 molecules. So that is what was asked in this question, formation of 2 moles of CO2. We can see 1 mole of CO2 is being converted to 2 moles of CO2, and in this process you have the generation of what other things, 1 moles of ATP, 3 moles of NADH and 1 moles of FADH2. Now let us look in this option. Do we have 1 mole of citrate formation in the output? No. So this option was definitely wrong. The second option says 1 mole of NADH formation, but from this cycle we can see that we have 3 moles of NADH formation. So again, that is why the second option is wrong as well. Third one is 1 mole of FADH2 formation. Definitely we have our 1 mole of FADH2 formation, which was our correct option. And the last one, 1 mole of oxaloacetate. Obviously this is not the end product, so this is also not found in the output substrates, output products. So the only correct option was option number C. So this is clear. Let us move to the next one. It says the 2 moles of CO2 produced in the first turn of the citric acid cycle have their origin in what? So they are talking about 2 moles of CO2 produced. We can already see in the output, this is the stage 2 only has been showed here of the Krebs cycle, but we can see the output is the formation of 2 CO2 molecules, 2 moles of CO2 has been produced. And in the first turn of citric acid cycle, what are the carbon? That means these carbons, suppose this is 2 CO2 molecules. So where are these carbon atoms coming from in the TCA cycle? That is basically the question. So this is your options. We know oxaloacetate is actually involved in the formation of 2 CO2 molecules. We have just seen it, I think. Yes, in this question we have just seen it, that from oxaloacetate we have this TCA cycle where we have the formation of 2 moles of CO2 molecules. But the question is, during this transformation, where are these carbon atoms coming from? So let us go back to that question. Yes. So what should be the correct answer? If you know it, you can write it. So let me show it. So these are the processes in the initial TCA cycle from which CO2 has been generated. So first of all, you have the reaction of acetyl-CoA with oxaloacetate. And what does acetyl-CoA does here? It transfers these acyl groups, acetyl group to this oxaloacetate, and you have the formation of this citrate. So this is marked in pink. So these pink colored carbons are coming from acetyl-CoA. It is not the inherent carbon atoms of oxaloacetate. Fine. So these pink ones are coming from acetyl-CoA. And we have our citrate. Now let us look at the next step. One step is missing here, the citrate to isocitrate conversion. I am not considering. I am directly going to the next step where we get our CO2 molecule. Now let us see. This is the step where we get our CO2 molecules. So now we have our isocitrate. We have isomerase working on citrate, giving us isocitrate. Now this isocitrate is forming alpha ketoglutarate and we have our CO2. Now look carefully. This COO minus, which is marked in blue coloration, that is forming the CO2. So this CH2COO minus that you are seeing here was coming from acetyl-CoA. If you look here, we can already see that this CH2COO minus at the top was coming from acetyl-CoA, but the other COO minus groups that we can see, that were already the integral parts of oxaloacetate. These COO minus groups are the parts of oxaloacetate. So these are the C minus that are belongs to oxaloacetate. Now if we look, one of the COO minus of oxaloacetate is converting to CO2 in this step that is shown here. Okay, and in the next step, after it gets converted to alpha ketoglutarate and CO2, so now you have again your alpha ketoglutarate here. One CO2 molecule from oxaloacetate has been formed. Next part, again if you see here, this upper part was from acetyl CoA, but this second COO minus group that is shown here, it is basically this COO minus group. Fine. So again this one was again a integral part of oxaloacetoacetate, which is again being converted to CO2 here. So both of the COO minus that were converted to CO2 were originally generating from or originally coming from oxaloacetate, not from acetyl CoA. So which carbon atoms are generating that? The two carboxyl groups. So let us look at the option. The first one says carboxyl and methylene carbon of oxaloacetate. So these are the carboxyl group and this one is your methylene carbon of oxaloacetate. Does this convert to CO2? No, we have already seen two COO minus group in each of these steps are actually converting. So this statement is a false statement. Methylene carbon does not convert to CO2. Next one is carboxyl group of acetate and a carboxyl group of oxaloacetate. So we have seen the COO minus group that was coming from acetyl CoA or acetate group that was coming, acetyl group was not involved for CO2 conversion. Only the oxaloacetate COO minus were. So this was again a false statement. The third one is carboxyl group of acetone and the keto group of oxaloacetate. Let us see. No, that was not the case. First of all, from the acetate no conversion was happening to CO2, and next one is the keto group of oxaloacetate. That means this CO group. No, this was not transformed, because if you look here, this is the CO group that they are talking about. This has not been converted to CO2. So again, this is a wrong statement. And the final one is two carboxyl group derived from oxaloacetate. Absolutely, we can see these two carboxyl groups are actually being converted to CO2. So the fourth option was our correct option. Fine. So let us move to the next one. It says, which among the following is or are part of coenzyme A structure? These are the option: phosphorylated ADP, markaptoethylamine, and third one is pantothenic acid. You have to identify which of them are a part of the coenzyme A structure. If you know it, please write it down in the chat box and then I will discuss. Okay, so for that we first of all need to know the structure of coenzyme A. So this is the structure of coenzyme A. Now what are the parts that you can see? Let us first look up to here. What is this? Adenine. And what is the thing? Phosphorylated, you can see. And how many phosphate groups are there? 2. That means what is this? ADP, this part only, and one of the OH group of this has been phosphorylated. Generally we have OH groups in both of the places, but one of the OH group here has been phosphorylated. What does that make it? Phosphorylated ADP. Originally it was adenosine dinucleotide phosphate, but after phosphorylation it has become phosphorylated ADP.   So yes, phosphorylated ADP,   ADP,    ADP,    ADP,    ADP,    ADP,    ADP,    ADP,    ADP,    ADP,    ADP,     ADP,    ADP,    ADP,    ADP,    ADP,    ADP,    ADP,    ADP,    ADP,    ADP,    ADP,    ADP,    ADP,    ADP,    ADP,    ADP,    ADP,    ADP,    ADP,    ADP,    ADP,    ADP,    ADP,    ADP,    ADP,    ADP,    ADP,    ADP,    ADP,    ADP,    ADP,    ADP,    ADP,    ADP,    ADP, So this is our phosphorylated ADP, which is a part of coenzyme A. Now let us look at the other parts. What is this? If you cut down here, what will you end up with? A molecule that is this. What will be the name of this? This is basically your markaptoethylamine. You have your SH groups here. Instead of OH, you have SH. That's why it is markapto. And this is your ethyl, 2 carbon atoms, and this is the amine group. So this part is your markaptoethylamine. So this is option number 2 is also a part of coenzyme A. And the last part, that is pentothenic acid. Now if you look the structure of pentothenic acid, so after this breakage you will have a COOH group here, and if you break here, actually you have to break here, then at this side you will have OH, and in the middle this group will be fixed. So after doing that, the structure you will be getting is the structure for pentothenic acid. So this is your pentothenic acid, and this is your markaptoethylamine. So yes, all of the three that were given in the options are the part of coenzyme A structure. So the correct option for this should be option number A. Okay, let us move to the next one. This is your next question. It says glycolysis leads to the production of what? First question is that. So glycolysis is what? It is the oxidation of glucose, and what does it results in? It results in the formation of pyruvate in normal condition, right? Nothing has been mentioned here. That means in presence of oxygen, glycolysis leads to the formation of pyruvate. Fine. And 2 molecules of ATP. At the beginning of the session, I have written down the equation involving glycolysis. There you can see we have formation of 2 moles of pyruvate along with formation of 2 molecules of ATP. Next is, in the absence of oxygen, fermentation leads to the production of what? The second blank that we need to fill. We have discussed this question as well. So what was fermentation? Involving glycolysis as well as the oxidation of NADH to NAD+. Together we call them fermentation. And when this happens, when you have deficiency of oxygen in your muscles, and that lead to the formation of or conversion of pyruvate to lactate, or lactate sometimes get actually the formation of lactic acid. That's why we feel the muscle fatigues because of the production of lactic acids. So in anaerobic conditions, that is absence of oxygen, the formation leads to the formation of lactic acid. So the first one is pyruvate, and this one was pyruvate, we all know, and second one is lactic acid. So up to this, if you know, you can already distinguish the correct option. What should be that? Option number B. Even if you just know these two, you should be able to answer this question. But there are other 2 options. Let us see that. Third statement is glycolysis plus the citric acid cycle can convert the carbons of glucose to what? So in glycolysis we have pyruvate, and that pyruvate goes into the TCA cycle or Krebs cycle. And what do we get from Krebs cycles? CO2. That means it converts the carbons of glucose to finally formation of CO2. So this third one should be CO2, and that absolutely matches with our options that is given in B. Next is and that stores the energy as ATP. You all know that. There is another blank, that is electrons are transferred from oxygen to water. So what are the electron transfer agent? In case of transferring, in the final step you have the formation of water molecules, but in the process which are the compounds that acts as electron transfer agent? You know that obviously NADH. Out of these options, the electron transfer agents are basically your NADH and FADH2. These are the electron transfer agent. In the last session we have learnt about this, that how many electrons they take up, 2 electrons reaction in one step, or two different steps, we have discussed about those. So those were the electron transfer agent, and in this case the agent is nothing but your NADH. So option number B should have been the correct answer for this question. Let us move to the next one. Okay, in this question it says conversion of fructose 1,6-bisphosphate to glyceraldehyde 3-phosphate is catalyzed by which of the enzymes that are given. So you have your fructose 1,6-biphosphate, and through which you are forming glyceraldehyde 3-phosphate. This is one of the most important step in the glycolysis. Basically, from fructose to glyceraldehyde 3-phosphate means there is a reduction in the number of carbon atoms. Fructose means there is 6 carbon atoms, but then you are moving to glyceraldehyde 3-phosphate. That is, glyceraldehyde means you have your 3 carbon atoms. Even if you do not remember all the cycle steps, still you should be able to answer this, because what are the enzymes that are given in the option? Aldolase, first one. Second one is hexokinase, C is enolase, and fourth one is phosphoglycerate kinase. First of all, you know, basically you are going from C6 to C3. That means some kind of cleavage must have occurred in the fructose 1,6-bisphosphate. But hexokinase and phosphoglycerate kinase, what are their role? Kinases means basically they transfer a phosphate group, so they cannot be involved in the cleavage process. So option number B and D are automatically rejected. Now you have your aldolase and enolase. So let us look at, this is the step for glycolysis. First step, you have the formation of glucose to glucose 6-phosphate. Basically, one phosphate group transformed from ATP to ADP. In the next step, you have the isomerization, which result in the formation of glucose to fructose. So glucose, basically it was a aldose, and from that you are going to fructose, that is a ketose. That is all that happened in this isomerization step. And this fructose 6-phosphate then again takes up another phosphate group, forming fructose 1,6-bisphosphate, because now instead of one it has 2 phosphate groups. Now what need to happen? If you see the structure, this is the structure for fructose 1,6-bisphosphate. Now you need to cleave it in this position. Why? Because ultimately we are going to form pyruvate. So we need reduction in the number of carbon atoms. That means some kind of breaking of bond is required. And which enzyme is coming to take place in this? It's nothing but your aldolase. So what is its function? The reaction is aldol cleavage. You can see in this structure, this is what? This is an aldol. You are cleaving it. So what enzyme will be involved here? Obviously aldolase. If it was an enol, then in that case you would have involved the enolase. But this is aldol. So obviously for aldol cleavage you will require aldolase, and that breaks into 2 forms. This 3 carbon forms this dihydroxyacetone phosphate, and the other 3 carbon form this glyceraldehyde 3-phosphate. So the correct answer for question number 10 should have been option number A. Okay, let us move to the next one. So it states, which among the following statements about glucokinase is false? So glucokinase is what? Even in the options, actually all the things regarding glucokinase is written. So let us just go back. We see this step, the conversion of glucose to glucose 6-phosphate, that is generally that is mediated by hexokinase. We can see this step. There is another enzyme that can do this step, that is basically your glucokinase. So apart from hexokinase, there is another enzyme that can form this glucose 6-phosphate, that is your glucokinase. What do you find it? You can find glucokinase in your liver, and it has a high Michaelis Menten constant or Km value for glucose. That means it has a high binding tendency with glucose. So where you can find it? In case of our liver. So the first statement is absolutely true, that glucokinase is found in our liver with a high Km value. And obviously it's a variation of hexokinase. It has to, because both of them are performing the same reaction. Both of them are converting glucose to glucose 6 phosphate. So obviously glucokinase has to be a variant of hexokinase because the role are same ultimately. So the second statement is a true statement as well. So this was true, this was true as well. Third one is liver glucokinase is subject to inhibition by glucokinase regulatory protein. Now let us go back here to discuss it. Although this is a bit out of topic, but let us discuss. So we can see we have our initial conversion from glucose to glucose 6 phosphate through hexokinase. But what is the problem here? We need a regulation. Why so? Because once it goes to glucose 6 phosphate, you can see this process is an irreversible process. So once the conversion has been happened, that means the other metabolic step will go forward. That means all of the glucose that is there in our body will be glycolized, but that is not what we want. We need some as storage for other situations as well. Not all the glucose that we take up, we need their dissociation and the energy formation, right? We need to store them as well. That's why we need regulation of these enzymes. So how do we do that? How can we do that? In case of hexokinase, we can either do it via a competitive inhibition method, where otherwise a feedback inhibition method. So what is this? Basically, in case of hexokinase, the formation of glucose 6 phosphate itself inhibit the reaction. We have learned about the inhibition or feedback inhibition, where the product that you get at the end of the reaction actually inhibit its formation, and that is how you regulate the overproduction of some product. That was true for hexokinase. But in case of glucokinase, that is not the case. Glucokinase is also converting glucose to glucose 6 phosphate, but this formation is not inhibiting the activity of glucokinase. So in this manner, hexokinase and glucokinase are different. But again, you still need the regulation of the formation of product, because in liver also you do not want all the glucose molecule to break down. So in case of glucokinase, the regulation is done by another protein known as glucokinase regulatory protein. It is not like hexokinase. It is not like hexokinase's feedback inhibition, but it is done by another protein, glucokinase regulatory protein. So the third statement was also a true statement. Now let us look at the fourth one. It says the ratio of glucokinase to glucokinase regulatory protein or GKRP remains constant in different metabolic states. So what will happen if the ratio of glucokinase to GKRP remains constant? So basically it is regulating your glycolysis of glucose. But at different metabolic state, you will not have the same concentration of glucose in your liver. Obviously, at different states, it will be different, the concentration and everything. So, if at every condition, metabolic states, GKRP tries to regulate it, then it will be problematic, because say the amount need to be certain. It cannot be very small, the glycolysis cannot be very limited, or it cannot be very high. It has to be some intermediate one. But if the ratio of glucose to GKRP remains constant no matter the amount of glucose present and no matter what is the metabolic state, then there will be no regulation between this. Whenever GKRP will see that there is glucose or the formation of, whenever GKRP will see glucokinase interacting with glucose, it will stop the reaction. But we do not want that. We need a regulation. That means the ratio of this will never remain constant. It will vary depending on the metabolic state and depending of the concentration of your substrates. So the fourth option that was stated here, that it remains constant in all states, is actually a false statement. So the correct option for question number 11 should be option number D. Let us move to the next one. Fine. This one state, which of the following is or are true about glycolysis? Fine, let us see. First is glycolysis takes place in the cytosol of cells. Absolutely, if we do remember the diagram, glycolysis originally happens in the cytosol, whereas the Krebs cycles occurs inside the mitochondria. So the first statement is a true statement. It definitely occurs in the cytosol. The next one is glucose enters the glycolysis pathway by conversion to glucose 6-phosphate. Yes, we have just seen the cycle. Let me go back to few slides. Yes, this is the glycolysis cycle where you can see the very first conversion or the very first step involved in glycolysis is this. The very first step is glucose to glucose 6-phosphate. That means this option where it's stating glucose enters the glycolysis by conversion to glucose 6-phosphate is another true statement. And the next one is glycolysis is a metabolic pathway that requires oxygen. Now if you see the overall process, so let me go back to the cycle. I don't think I have written it here. No, it is not here. This is not also the overall glycolysis process. So let me just state it. So in the overall glycolysis process, you have the conversion of your glucose to pyruvate and you have the generation of your H2O molecules. But if you look throughout the process, it is not necessary that oxygen molecules needs to be present. Why so? First of all, we know glucose breakdown or glycolysis will occur in both aerobic condition and anaerobic condition. In aerobic condition, we will have our pyruvate and that will go into our Krebs cycle, but in our anaerobic condition, we will basically have the conversion from pyruvate to lactate. But in none of the steps, we do not involve direct oxygen molecules. Even at the end of the glycolysis, you know one molecule of H2O is being produced in the overall process. But in that step also, if you remember, basically in that step, from 2-phosphoglycerate to I do not remember exactly, from 2-phosphoglycerate we have the transformation that is done by enolase, and that step you have the reaction between, let me go back here, you basically have the reaction between H plus and OH minus, which is governed by a enolase intermediate, and that results in the formation of H2O. That is also not coming from a oxygen molecule. No, this conversion is not taking from a oxygen molecule. It is individual H plus and OH minus form an enolase intermediate, guided by enolase enzyme, you have the formation of H2O. So in none of the steps you have direct requirement of oxygen. So the last statement was actually a false one. So accordingly, so which 1 and 2 should be the true option and B should be the correct option for question number 12. Let us look at the next one. Okay, I have just discussed in a few slides back Glucose 6-phosphate inhibits hexokinase enzyme by what mechanism? So we all know by now that glucose is being converted to glucose 6-phosphate by hexokinase. But we do need a regulation, and how the regulation will happen? First of all, one thing, so we have our glucose to glucose 6-phosphate and we have our hexokinase. So how can we regulate this enzyme so that unnecessary amounts of glucose 6-phosphate has not been formed? Okay, so how do we do that? First of all, we know that their product itself, glucose 6-phosphate, is actually inhibits this reaction. So what kind of inhibition is this? That is your one kind of feedback inhibition. And in the option you can see, so how does this feedback inhibition works? If you do remember, in case of feedback inhibition, what happens? You have your G6P. So this was the enzyme, suppose, and from it you now have the product. But once this product is getting released, it is somehow altering the conformation of this enzyme. After the product has been released, suppose the active site now looks like this. That means some kind of conformational change has been occurred within these enzyme molecules. Because of that, suppose it was enzyme, it was our substrate, and this is the overall reaction. Now because of this conformational change, this can no longer take a substrate that looks like this, right? So obviously our production of G6P will be less. Now it will be in control because of this inhibition process. So one of the inhibition is feedback inhibition, where we have a allosteric interaction. This kind of conformational change where the product while leaving makes a conformational change in the enzyme is known as allosteric interaction. So this is one of them. Another way also applies, that is hexokinase can also inhibit the reaction via a competitive pathway. So how does the competitive pathway occurs? So suppose, let me draw the product like this. Okay. Suppose the product looks like this. So if both of them can fit in this site, so they will compete with each other. Basically they are fighting for one chair. So in that way as well, the inhibition will occur to some extent, because the substrate can now also sit. That means your glucose can also sit at the enzymatic site, and the glucose 6-phosphate can also sit at the enzymatic catalytic site. So both of them will compete for that place, which will again lead to the regulation of the formation of product. So these are the two interactions or two inhibitions methods through which hexokinase acts on glucose 6-phosphate. So the correct option should be which one? Competitive inhibition and allosteric interaction. So option number C is the correct option. Let us move to the next one. It says the given figure shows the TCA cycle or the citric acid cycle. So you need to fill up this A and B. I have already shown you the entire TCA cycle. Let us go back here. So this was our initial citrate to, we have our isocitrate to and which enzyme acts here? Your aconitrase, which acts here to form this citrate to isocitrate. Next step is isocitrate to what? That is your question. So in the next step we have the formation of isocitrate to alpha ketoglutarate. So the next one, this particular one is alpha ketoglutarate, that is governed by the enzyme alpha ketoglutarate dehydrogenase. Then we have the formation of succinyl CoA followed by succinate, fumarate and malate. Again in this step we will have the action of malate dehydrogenase that will lead to the initial substrate of TCA cycle, which is none other than oxaloacetate. Because we do remember oxaloacetate will react with coenzyme A, acetyl CoA. Basically it will react with acetyl CoA and that will ultimately result in the formation of citrate. So what was our A? A was alpha ketoglutarate and B was oxaloacetate. So the correct option for this should be option number C. Okay. Let us move to the next question. Again the same TCA cycle has been given here. It is saying if I is the enzyme required to convert citrate to isocitrate. Okay. So we have the conversion of citrate to isocitrate. This is termed as enzyme 1. And the second one is 2 enzyme is required to convert malate to B. Then identify 1 and 2. I have already discussed while discussing the previous question. So what enzyme acts here? It's your aconitase. So basically citrate to isocitrate is governed by the enzyme aconitase. So our one enzyme is nothing but aconitase. If you know that, you already know which option should be correct. Still, what enzyme is used here? From malate to we are forming oxaloacetate here. And which enzyme governs this reaction? This is nothing but you have malate to oxaloacetate. So the enzyme that works here is malate dehydrogenase. So which should be the correct option then? Option number B. 1 is aconitase and 2 is malate dehydrogenase. Option number B. Okay. Going to next one. It says in an eukaryotic cell, most of the enzyme of the citric acid cycle are located in what? So we just said that the glycolysis process occurs in the cytosol of the cells, but the Krebs cycle or the TCA cycle occurs within the mitochondria. So if it occurs within the mitochondria, then where should the enzymes lie? Obviously in the mitochondrial matrix you have the maximum places or maximum enzyme that will lie in the mitochondrial matrix where the Krebs cycle basically takes place. So the correct option for question number 16 should have been option number B. Fine. So this is the next question. It says a student identifies a new enzyme malase, stating it has a function that is opposite that of fumarase on fumarate. So for that, what may be the possible function of this enzyme malase? So there, suppose someone has discovered a new enzyme and it has named it as malase. Now they are saying we already know about a reaction fumarate that transfers into some substrate through the action of this enzyme fumarase. If we do know the reaction, then the student is saying malase does the opposite one. So basically we should have known that what is the work of fumarase. So what does it does? This is the structure of fumarate that I am showing. What does it do? It hydration of fumarate to malate. You can see here is a double bond. Fumarase does a hydration reaction, due to which you can see there is a addition of OH molecule to this carbon atom and the hydrogen from this H2O to this carbon atom, resulting in the formation of malate. So that is the reaction. Now according to this student, malase does the opposite. So what should be the opposite? There should be a dehydration reaction where H2O molecules will get out from the system and you will end up with a double bond here, because if these two comes out you will have a double bond here. That should be the reaction. So what should we name it? That should be malase dehydrate malate to form fumarate, that is the opposite reaction, malate to fumarate. And what is this? This is a dehydration reaction. So A, option number A should be the correct answer for question number 17. Okay, let us move to the next one. So it says the very first step of the glycolysis after the polysaccharide is broken down to glucose uses the enzyme H to phosphorylate the glucose molecule. So again we should already know. So glucose is entering in the for the first step of glycolysis. And what is the first step of glycolysis? The formation of glucose to glucose 6 phosphate. And which enzyme takes place for the phosphorylation? Here it is nothing but your hexokinase. We have already discussed about it so many times. So the correct option for question number 18 should be option number A. Okay, let us see this one. I have already showed still. So this question says electrons are extracted from the citric acid cycle for use in the electron transport chain. Okay, through what? So this is the question. We have discussed it in some of other questions previously as well. So you have to basically identify which among them works as electron transport chain. So where from which of these species you get your electrons. So we already know what are the electron transporters here. We have our NAD plus that will take up 2H plus and 2 electron leading to formation of NADH. So this is one of our electron transporter. Another one is FAD, which will take again 2H plus and 2 electron in 2 consecutive steps, resulting in the formation of FADH2. So the correct option should be the reduction of NAD plus and FAD. These are the electron transporters from which you get the electrons for TCA cycle. So let us move to the next question then. Try to match the followings. We have to match them. So in the left hand side we have the enzymes isomerase, dehydrogenase, synthase and mutase. You have to match them with their functions on the right hand side. So for the enzymes, from their name itself, it is very clear that what is their role. So what does an isomerase do? First of all, if anybody knows, please try to do it by yourself. This one is very easy and I will discuss it in a minute. Try to do it by yourself and then I will show you the answer. If anybody can match this, please feel free to write your answer and then I will show you. Okay, fine. So from the name itself, isomerase. So what should it do? It will form an isomer of the substrate, obviously. So where should it go? So this is our isomerase. It should connect to 4 converts one isomer to another. So basically the conversion between isomers happens through isomerase. The next one is dehydrogenase. Again, from its name itself you can understand what will dehydrogenase do. It will remove hydrogen. Dehydrogenase means dehydrogen. That means you are removing hydrogen. So obviously removing hydrogen will lead to the oxidation of any species. So obviously dehydrogenase should match with function 3. Next one is synthase. What does a synthase do? From the name itself you again know. Synthase will combine 2 things to make a new compound. Basically it will synthesize a new compound. So enzyme C matches with function 2. The next one is mutase. What does a mutase will do? Mutase basically a group transfer enzyme where it will move the phosphate groups from one position of the sugar to another. So this should lead to function number 1. So how does that look? If you have your C1, C2, C3 etc. I am not drawing the other functionalities, and suppose you initially had your phosphate group here. Here suppose you have your phosphate group. Fine. Mutase will function in some way that suppose it is transferring to the second carbon atom. Only this much. Transferring the phosphate group within the sugar molecule from one carbon to another. So that is the work of mutase. So what do we have now after matching? Our A matches with 4. Our B matches with 3, followed by C matches with 2. So obviously D will match with 1. So this is the combination. According to that, the correct option should be option number D. Fine. Let us move to the next question. So in this question it is stating that given are the advantages of M. What is M? That is not stated. So let us look at the advantages. First of all, first advantage is higher rate of reaction, because product of one catalyzed reaction acts as a substrate of another reaction. It is also available for the active site of another enzyme without much diffusion. Fine. Another advantage is minimum site reaction, and third one is coordinated control. Now you have to identify what is M. These are your options: a single enzyme system, a multiple enzyme complex system, parallel reaction system, or an oscillatory reaction system. These are your 4 options. So let us see. First of all, if you remember the overall glycolysis process, so this is the overall process that is shown here in 2 different boxes. Now you know, in case of glycolysis, once this 6 member, up to this point we have our 6 carbons. After this, through the act of aldolase, we have now 2 C3 parts. One is glyceraldehyde 3-phosphate and another one is dihydroxyacetone phosphate. Now these these two compounds or these two products are actually isomer to one another. One is ketone form and another one is aldehyde form. So basically these two are isomer. But out of these two, only glyceraldehyde 3-phosphate can further continue the glycolysis process. This dihydroxyacetone phosphate cannot continue in the glycolysis process. But because they are isomer, both of them will be formed, and this transformation between these two will be governed by some kind of isomerase. You know, already know that isomerase do the conversion between two isomer, helps in the process. So try this sorry, this triose phosphate isomerase is basically that particular isomerase which governs the isomerization between these two compounds, this ketone compounds and this acetaldehyde compound. But this triose phosphate isomerase has its affinity in somehow that it generally produces or generally converts most of the molecules to this dihydroxyacetone phosphate. Okay, so it has higher tendency to form this one, but we already know that dihydroxyacetone phosphate cannot continue the glycolysis process. Only the glyceraldehyde part will do. But now the advantage is, although suppose I am just saying for an example, suppose this isomer is present as 10%, but this one is present as 90%. because of the action of this triose phosphate isomerase. Fine. But as soon as this glyceraldehyde 3-phosphate is being formed, in the next step it will be used by this glyceraldehyde 3-phosphate dehydrogenase, and this process is very fast. Very quickly this glyceraldehyde 3-phosphate will be taken out. So as soon as this will be taken out from the system, to maintain the equilibrium, now more dihydroxyacetone phosphate needs to be converted to this. So initially it was 90%, 10%, but as soon as this 10% has been formed, it is immediately taken up by other enzymes for the next process of the glycolysis. To maintain this equilibrium, now more number of dihydroxyacetone phosphate needs to be converted to glyceraldehyde 3-phosphate. Now this only happens because of this next step of enzymatic reaction. If this was not taken up by this enzyme, then the conversion or the shifting of the equilibrium towards this was never necessary. This is only happening because there are many enzyme systems involved in the overall process. So that is why you are getting the higher rate. Because of this reaction, because of this multiple enzyme involved reactions, the overall glycolysis is happening. So what has been stated in 1, that higher rate of reaction occurs because of the active site of another enzyme, presence of available active site of another enzyme, which is obviously true. In case of this, because of the presence of glyceraldehyde 3-phosphate dehydrogenase, you have the conversion of this to in a faster rate. So this happens because of involvement of multiple enzymes. So what should be the correct option? If you know now already, and this is minimum side reaction obviously because it generally involves enzymatic reaction. So you have minimal side reaction and you will have your coordinated control. So what do you mean by coordinated control? Because the conversion is very fast in this step, the equilibrium will also shift in this direction because it is very easily taken up. So the equilibrium is also shifting in the left hand side here as well. So this is the coordinated control. The control or the concentrations of these substrates or the products are being controlled in a coordinated manner. So this all results in case of multi-enzyme complex system. For a single enzyme, this would not have been possible, but because of the involvement of multiple enzymes, this is being possible. So the correct option for 21 should be option number B. Moving to the next one. Okay, so this one we have already done, so I will not spend much time in it. So it says if the given table describes the pyruvate dehydrogenase subunits, fill in for A, B and C. So we initially saw that pyruvate dehydrogenase has 3 subunits that are denoted as E1, E2 and E3. Now if you do remember their structure, so for the first one, pyruvate dehydrogenase, in the prosthetic group we had our TPP or thiamine pyrophosphate. In the second one, this one E2 part, okay, we have the next prosthetic group which is lipoamide. We initially did that lipoic acid was involved in the reaction and it is formed via lipoamide. And the third part is E3 subunit. In case of that, we had the third prosthetic group, that is nothing but NADH. So these are the prosthetic groups that are involved with the 3 subunits of pyruvate dehydrogenase. So accordingly, the correct option should be option number C. Let us look at the next one. So it says glucose 6-phosphate is formed from glucose with the help of a certain enzyme. We already know the enzyme, that is hexokinase. But glucose 6-phosphate inhibits the action of this enzyme. Obviously that is hexokinase. Then, so we already know the enzyme in the first two lines that we are talking about is nothing but our hexokinase. The next statement is G is a variation of this enzyme that is found in the liver. So what is the variation of hexokinase that we can find in liver? It is obviously glucokinase. We have discussed this in details. And glucokinase, that is our G, that has a high Km value or a high Michaelis-Menten constant value towards glucose. Also, it does not get inhibited by the product. We know the control for glucokinase comes from glucokinase regulatory protein or GKRP. It is not the allosteric or competitive inhibition with the product. So obviously G should be glucokinase. We have already discussed, so this should have been the answer. Moving to the next one, it says the given figure illustrates the action of a certain enzyme. The enzyme required for the reaction and the product 2 are respectively what? So what do you need to identify? A conversion is happening from 1 to 2. So first you need to identify the enzyme that you require here, and you have to identify this molecule, the 2 molecule that has been forming. So what we can see, if we see here what is the change that is happening, from aldehyde group we have addition of a phosphate group here now. So this is our inorganic phosphate that has been added to this aldehyde group, giving you this bisphosphate molecule. So what should be the name of this compound? 1, 2, 3. So 1, 3 bisphosphate glycerate. That should be the name, obviously, because if you know it is 1, 2, 3. So at 1 and 3 position you have your phosphate group. So 1, 3 bisphosphate, and this is a basically the basic structure is glycerol. So from that we have C double O minus, so it is our glycerate. So the substrate is glycerate, and what enzyme should take place? This glyceraldehyde 3-phosphate dehydrogenase. That is the reaction. Why it is saying that? Because ultimately it is an oxidation reduction reaction. Why? Because you can see NAD plus is getting reduced to NADH in this process. So this is a reduction process. If NAD plus is getting reduced, so the substrate must be oxidized. That is the main thing for any redox reaction. So in this kind of oxidation reduction reaction involving hydrogen, who will come to play the role? Some kind of dehydrogenases that will come here, because ultimately this hydrogen group from this aldehyde moiety has been removed, and you have having a formation of a glycerate molecule. So obviously some kind of dehydrogenase must have acted here to remove this hydrogen molecule. So the name of this compound is glyceraldehyde. You have 1, 2, 3. So this was your glyceraldehyde, but at 3 position you have phosphate. So the name of this one molecule compound is glyceraldehyde 3-phosphate, and its dehydrogenation is taking place. So the enzyme that is working here is basically glyceraldehyde 3-phosphate dehydrogenase. So just if you look at the structure of the molecules, it is actually very easy, even if you don't know the answer, but from the action that is being taken place and from the basic name, it is very easy to identify the enzymes and the resulting substrates. Okay, moving to the next one. Fine. So it says for the given enzyme at the active site there is an X-thiol which plays a major role in the catalysis. The reactant for the enzyme catalase reaction reacts with this thiol to form Y intermediate. What is X and Y? So what are we talking about? Just we discussed about this reaction in the previous slide, but we directly showed this step. But there are multiple steps involved between the transition and the enzyme that comes into play, glyceraldehyde 3-phosphate dehydrogenase. there are multiple steps that are involved. So that we did not show. So let us look at the steps. So first of all, there is a thiol that is involved here. What kind of thiol? The cysteine SH group of the enzyme, the glyceraldehyde 3-phosphate dehydrogenase, the enzyme that we talked about, its cysteine SH group or cysteine thiol group takes the first step of this reaction of this dehydrogenases. So the SH of cysteine is reacting, so we already know. And what is it forming after attacking at the aldehyde side? Now we have this one, the formation of this one. What is this? This is a thiohemiacetal. Generally, if it was a OH, we would have called it a hemiacetal, but instead of OH, an SH group is interacting here, so that will be a thiohemiacetal intermediate. So what should be the correct answer? That the thiol group is coming from a cysteine moiety, and what intermediate is forming? A thiohemiacetal. So accordingly, the option number A should have been the correct answer of this question. Okay, let us move to the next one. Okay, so we have this diagram. In this diagram, we have the transformation of glucose to, in the final step, half of the glycolysis is actually been shown here, from glucose to glucose 6-phosphate, followed by fructose 6-phosphate, followed by some formation which is termed as X, followed by the formation of glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. So what do we have here? We need to identify our E2, this one, and X. This we need to identify. So first of all, let us look at this. What is this? Glucose to glucose 6-phosphate, we have our E1. What is E1? We have our hexokinase. We have discussed this multiple times. So now we have our glucose 6-phosphate. It has been converted to fructose 6-phosphate. So what is this conversion? Basically, from a aldose, you are going to a ketose. This is also C6, this is also C6. No other change has been occurring. So what type of reaction is going in this case? That should be an isomerase reaction, because you are transforming an aldose to ketose, basically two isomers. So E2 is nothing but your phosphoglucose isomerase. What do you have here? A phosphate that is attached with glucose, so it will be termed as phosphoglucose. So we have our phosphoglucose, and that has been isomerized to fructose. So the enzyme is phosphoglucose isomerase. Okay, so we have identified E2. Next is E2 is forming some compound X. What should be the next step? We have our fructose 6-phosphate. Now look here, ATP is being converted to ADP. That means one phosphate group is removing from here, right? If ATP has been converting to ADP, that means one phosphate group is leaving. That phosphate group should attach where? To this fructose 6-phosphate, and what would will result in? Now instead of one phosphate group, we will have two phosphate groups. So that will result in the formation of fructose 1,6-bisphosphate. In the 6th position, we already had one phosphate, another addition at the first position. So that will end up in the formation of fructose 1,6-bisphosphate. So we have this, and finally this fructose 1,6-bisphosphate, we already know. Here we have the action of aldose, basically the cleavage of this aldol cleavage, resulting in two compounds having C3 carbon atom. That means 3 carbon atoms each. That is glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. We already know. So the correct option 26 should have been C. Okay. Question number 27 says the enzyme participating in the given reaction and the amino acid involved in its functioning is what? So what kind of reaction first happening we need to see? What kind of structural change has been occurred? You can see this part is fixed. only the phosphate group is being transferred from this C3 carbon to now to this C2 carbon. Only this much change has been occurred. These positions, only this C2 to C3 positions had been changed. So only a phosphate group has been changed within this molecule, but from one carbon to another. So what kind of enzymes acts like this? Mutase. So if you now look at your options, you only have one options that have mutase. So what enzyme should be that? So obviously you have your 3-phosphoglycerate. So that should be converting it to 2-phosphoglycerate. So the main moiety is same, phosphoglycerate. So the mutase should be named after that. So the mutase is phosphoglycerate mutase. Very easy. From the name and the reaction itself you can tell it is phosphoglycerate mutase. And what is the enzyme's active site? What amino acid is sitting at the active sites of the enzyme that is helping in this reaction? So this is the reaction through which phosphoglycerate mutase transform the phosphate group, and at the active site you will be able to find histidine. In the enzyme's active site you can see histidine that helps in this inorganic phosphate groups transfer. Okay, so the correct option for this should be C. Moving to next one. So this is a numerical question. It says if the oxidation of one glucose molecule yields an energy of 2840 kilojoule per mole. Oxidation of one glucose molecule. Okay. And each ATP molecule contributes 30.5 kilojoule per mole energy. You already know that from 1 ATP you got 30.5. Now they are saying find the number of molecules of ATP necessary for 60% efficiency of the biochemical engine in living system. So what is our biochemical engine? How are we getting this energy? So basically you are breaking down glucose for the functioning of our biochemical engine. And how much energy does that require to break down glucose? You yield the 2840 kilojoule per mole, and each ATP gives you 30.5 kilojoule per moles energy. So how many ATP molecules you will require for 60% efficiency to your body to function at 60% efficiency? So let us look at that. How to solve that? You already know oxidation of glucose you have 2840. You don't know how many ATP molecules you require. Suppose we require X number of molecules. So we have our 30.5 into X divided by our 2840 overall glucose energy that we are getting, equal to 60% efficiency, that is 60 by 100. So if you solve this you will get the value of X is 56. So how many ATP molecules will we require for 60% efficiency is 56. Okay, that was how it should have been solved. Okay. Moving to the last question, I guess. Yes. So this is the last question. It states identify the correct statement. So the true ones we need to identify in case regarding metabolism. The first one is catabolism is the degradation of nutrients to generate energy and starting materials. Absolutely yes. That was the definition of catabolism, that you degrade your nutrients and generate energy. Basically the breakdown of glucose. So that is a kind of catabolism. If you divide it, metabolism, there are mainly two parts. One is catabolism and another one is anabolism. So catabolism is where you break down something to get energy. So this is a true statement. Next one is anabolism is the synthesis of biomolecules from starting material. Absolutely yes, this is just the opposite of catabolism. In case of catabolism, you are breaking down the biomolecules to get your energy. In case of anabolism, you are just doing the opposite. You are synthesizing the biomolecules from some starting metabolism. From some starting materials. So yes, the second statement is true as well. Third statement is catabolism and anabolism are opposite processes. We just said it now. So yes, this is true. And the fourth one is metabolism involves the usage of substrates, intermediates and products known as metabolites. Absolutely yes. In case of metabolism, that involves both catabolism and anabolism. We have our substrate from which we get to have many intermediates in between, which finally gives us product, and all of them, all of the intermediates, all of the products and the substrate that we have started with, all of them collectively known as metabolites. So yes, the fourth statement is also a true statement. So all of them are true statements, so accordingly the correct option should have been option number D. Okay, I think this was the last one. Okay, I think with this I will stop today's session, and this will be the last session. Thank you all for joining, and a very best of luck for your exam, and thank you for all the time that you give me. I'll stop the recording.